Good morning, all of you. Now, till now, we have discussed about the metabolism. I started with the topic of metabolism of amino acids. Yesterday, I discussed about the metabolism of phenylalanine, which I told you all it's an essential amino acid. Now, today we are going to discuss the metabolism of another essential amino acid, which is tryptophan. Now, I gave you a very basic concept regarding this metabolism. Whenever we have to memorize it also, it's very, difficult to memorize the metabolism of individual amino acids. But I'm giving you just a tip how to memorize it. First of all, you have to focus this thing in mind. Ki amino acid is it is an amphoteric substance, means it can behave as an acid as well as a base. So its metabolism is divisible into two categories. First is that toxic ammonia forms urea, and another part of metabolism is as per the carbon skeleton. So as per the carbon skeleton, that COOH group is actually involved, either it is catabolized or it is metabolized to form certain specialized products. And on the basis of this thing only, amino acids are categorized into glucogenic, ketogenic, glucogenic and ketogenic. Now this tryptophan, the major feature about this tryptophan is that it is a essential amino acid. Essential means, again, it cannot be produced in our body, it has to be supplemented from outside diet. Then this is, if we see the structure of this, it is heterocyclic in nature. Heterocyclic means it has an extra indole ring present in it. And this indole ring actually gives it altogether a different metabolism. And if we see in terms of, you can say as per this, it is glucogenic as well as it is ketogenic in action. A very important feature about tryptophan is 60 milligram of tryptophan is required to generate 1 milligram of niacin, which is nothing, which is a type of vitamin B complex. And the most important function of this niacin is its active form in the body is NAD, NADPH, the most powerful reductant in all the biochemical reactions. So these are the four very important points regarding tryptophan. Now, since I told you we have to study the metabolism as per the carbon skeleton, कि how this tryptophan is catabolized in the body and what are the important products synthesized by it. So if we see, this metabolism is actually divisible into two subheadings. One is, as per catabolism, it undergoes kynurenine anthranilate pathway. And second is the specialized products, which are serotonin and melatonin. So you can see in just one slide, I have depicted the entire this thing of metabolism of tryptophan. One is it undergoes degradation by kynurenine anthranilate pathway. This I am going to discuss now. And it forms specialized products, serotonin and melatonin. This melatonin is a precursor of serotonin. Again, these type of questions are generally asked in the exam. One word question. Precursor of serotonin is? It is tryptophan. Similarly, precursor of melatonin is also tryptophan. Now, first of all, we are going to take up this kynurenine anthranilate pathway. So, what happens is tryptophan, again, a amino acid. First process would be it has an extra indole ring present with it. If we see the structure of tryptophan, this is the extra indole ring which has to undergo degradation. So there will be splitting of this, forming N-formyl kynurenine. The enzyme is pyrrolase, tryptophan pyrrolase. Once this N-formyl kynurenine is produced, this formyl group has to be removed now because this is kynurenine enthranilate pathway. So by enzyme formylase, it will be given out in the form of format, and this N-formyl kynurenine will be converted into kynurenine. Once this kynurenine is produced, then it undergoes splitting and hydroxylation both to form 3-hydroxylkynurenine. The enzyme here is kynurenase. A very important reaction because it requires vitamin B6 as one of the major coenzyme. So in the presence of B6 pyridoxal phosphate, this undergoes splitting to form 3-hydroxylkynurenine, and this 3-hydroxylkynurenine, then it will give what? It gives out alanine, and it is converted into 3-hydroxylenthranilate. Now once this 3-hydroxylenthranilate is produced, this by various steps will be converted into niacin, or which is also known as nicotinic acid, and it is converted to form 2 molecules of acetyl coenzyme A. So this is how tryptophan undergoes degradation like this. Let's quickly see this. Splitting of the indole ring, forming N-formyl kynurenine. This N-formyl kynurenine will give out this formyl group converted to kynurenine. Kynurenine in the presence of vitamin B6 forms 3-hydroxylkynurenine, and this 3-hydroxyl, as you can see, it is giving this alanine. Alanine, which is again a precursor of gluconeogenesis. So you can say ki, here you can say tryptophan is glucogenic in action by giving out alanine in this step, and here you can see it is giving acetyl coenzyme A also. So it is what? It is glucogenic as well as ketogenic. Very, very important. Now let's take up this reaction. This is important again. In the deficiency of B6, what will happen? There won't be this reaction. There will be increased concentration of this kynurenine. So this kynurenine, instead of being converted into 3-hydroxylkynurenine, will be converted to form xanthurenic acid. And that is why we say excretion of xanthurenic acid in urine depicts B6 deficiency. Very, very important. In subjects, in persons who are deficient of this B6, there will be excretion of xanthurenic acid in urine, which is actually an indication of this. Right? So this is how tryptophan undergoes catabolism by kynurenine enthranilate pathway. And you can see from this entire picture, this tryptophan is undergoing degradation to form 1 molecule of alanine and 2 molecules of acetyl-CoA, so that it is glucogenic as well as ketogenic in action. Right? Then next is, this is one way of degradation of tryptophan. Then another is this tryptophan is required to synthesize serotonin and melatonin. Now what happens is, this tryptophan will undergo hydroxylation first. So the enzyme will be obviously tryptophan hydrolase. It is going to form 5-hydroxyl tryptophan. And once it undergoes hydroxylation, it gives out a molecule of carbon dioxide, forming a very, very important biogenic amine also, which is 5-hydroxyl tryptamine or which is serotonin. Again, very, very important. So, tryptophan by two-step reaction forms serotonin. Once the serotonin is produced, it undergoes again a two-step reaction forming melatonin. So, these are the two very important products which are synthesized by serotonin by tryptophan. One is this serotonin and another is this melatonin. Now all of us know this serotonin is what? It is a potent vasoconstrictor and it helps in stimulation of the cerebral activity. All the depressants, all the antidepressant drugs act on either degradation of serotonin or prolonging the action of serotonin. Now see, a very beautiful feature about metabolism is what? Nothing is being, you can say, whatever is there inside our body, it is not wasted as such. Whatever is produced, it is consumed by our body or it is converted into such, you can say, non-toxic substances which can be excreted in urine. Same is the case with serotonin. Once its function is over, this serotonin is degraded to form 5-hydroxyl-indole acetic acid. On average, there is excretion of 5 to 7 milligrams of this in urine. Degradation of serotonin takes place in the presence of a very important enzyme which is MAO or which is also known as monoamino oxidase. So all the depressant drugs, all the antidepressant drugs, they are going to either activate MAO. If MAO is activated, automatically there will be more and more degradation of serotonin, as it is clear from this diagram only, from this picture. And similarly, if they are increasing the action of serotonin, automatically they will be the inhibitors of MAO, like the action of reserpine and iproniazid. So this is how they are acting on this. And similarly, this is how serotonin is being produced and degraded. Let's take up this melatonin. This melatonin is a hormone which is secreted by the pineal gland. And this melatonin is required for the circadian rhythms in our body. Or you can say this melatonin is responsible for the sleep and awake processes in our body. Its production increases at night. So normally when we say that the diet, if we take a diet rich in proteins, like you can say, or you can say if we take diet rich in banana, or if we take tomatoes, if we take, you can say, high protein diet, which is actually having tryptophan in this, it is going to synthesize, it is going to increase the production of this melatonin. So this is how serotonin and melatonin are synthesized by tryptophan. And this is how we study the metabolism. Now, a very important feature about tryptophan, just now I told you, 60 milligram of tryptophan is required for the synthesis of 1 milligram of niacin. Right? Niacin, which is basically in active form, it is NADP or NADPH. Deficiency of this niacin can lead to pellagra also. And that is what exactly happens. If we are taking a diet deficient in this tryptophan, automatically there will be a deficiency of niacin in our body, because this is the only vitamin which is being produced by this amino acid. Now see, what happens is, in general, if we say 1% of serotonin is produced, and serotonin is actually very actively produced in the argentifine cells of the body. Now what happens is, during carcinoid syndrome, there is increased, or you can say tryptophan is diverted to form more and more of serotonin. In carcinoid syndrome, there is, you can say, overproduction of serotonin. On the other hand, what will happen is, the major symptom in carcinoid syndrome would be the person will be deficient of pellagra also. Why? Because tryptophan, normal concentration by tryptophan, only 1% of serotonin is being produced. But in carcinoid syndrome, the production of serotonin increases. And if the production of serotonin increases, there will be lack of production of this tryptophan to niacin. So the person will suffer from pellagra. And this is how we have to, you can say, categorize the metabolism of a particular amino acid. First of all, we should know how it is catabolized to form different products. On the other hand, we should know what are the specialized products synthesized by this. And third part is what are the inborn errors of metabolism associated with this. So next we are going to take up, now this is clear. So overall, if we see the metabolism, we can say that tryptophan is a essential amino acid, heterocyclic in nature, and it is glucogenic as well as ketogenic. It is forming one molecule of alanine and two molecules of acetyl coenzyme A, and it is required for the synthesis of serotonin, melatonin and niacin. Then we come over to the next segment. What are the inborn error of metabolism associated with tryptophan? So the most important inborn error, first is obviously pellagra, which is characterized by 3 Ds: diarrhea, dermatitis and dementia, which is finally leading to the 4th D, which is death. Second is Hartnupp's disease. It is actually named after some British, you can say, legend, because it was inherited in his legacy. So that is why it is Hartnupp's disease. And basically what happens is there is defective tubular reabsorption. So there is excretion of indole derivatives also in this. So these are the two inborn error of metabolism associated with tryptophan. I hope it is clear to all of you. Then next metabolism which we are going to take up is about sulfur containing amino acids. Now there are two sulfur containing amino acids, cysteine and methionine. This methionine is considered to be the essential amino acid. Now why it is essential? Because methionine is required for the synthesis of cysteine in our body. And if we see the structure of methionine and cysteine, methionine, this is the generalized structure. I am not going to draw the generalized structure. I am going to write SCH3 here, and in this it is SH. So you can see the structural difference. This methionine has an extra methyl group which is lacking in cysteine, and this group is known as sulfhydryl group. Right. Now just now I told you methionine is essential, cysteine is non-essential, which means methionine has to be required, it has to be supplemented from the diet, and this methionine is required for the synthesis of cysteine. So now here we are going to concentrate on the metabolism of methionine. So the basic features are, obviously just now I told you it is essential. The most important function of this methionine is it has a extra methyl group, or it is known as methyl donor. And this type of reaction is known as transmethylation. Very, very important. Or in other words, we can say methionine can give rise to one carbon metabolism. One carbon means this one carbon is coming in the form of methyl group. So these are the important features of methionine. Now then we come over to the metabolism part. Now see, if we see the metabolism, this methionine, now every amino acid or every metabolite, whenever it has to perform a function in the body, it needs to be activated first. Always. Without activation, it cannot react also. So this methionine for activation requires ATP. So this is a very interesting reaction. In fact, ATP will come and it will give its adenosine group. See, ATP is adenosine triphosphate. Phosphate will be given out as such. Only adenosine will be there, and this methionine will be converted into SAM. SAM stands for S-adenosyl methionine, which is actually active methionine. This is the active methionine. Now, because it is ready to donate its methyl group, this SAM will obviously donate its methyl group. The acceptor is going to accept it and methyl group will be given. Once it donates this methyl group, it will be converted into S-adenosyl homocysteine. Now this S-adenosyl homocysteine, now what the function of methionine was to give only this methyl group. Once it has donated this methyl group, its function is over. It has to be converted back to methionine so that it can again give more and more of methyl group. So this S-adenosyl homocysteine will now give out this adenosine. Once it gives out that adenosine, it will be converted into homocysteine now. Now this homocysteine, now see, just now I told you methionine has this methyl group. It is donating this methyl group. So overall, if we see, this homocysteine is without a methyl group. Or in other words, we can say if we put a methyl group in homocysteine, it will be converted back to methionine, isn't it? This methyl group comes from folic acid. Folic acid, which has this methyl group, and it will be converted into THF, forming methionine back. Another very interesting feature about this is here B12 is required as the coenzyme. So you can see this we are going to discuss in detail also when we are going to discuss vitamins. Here you can see it's a one-step reaction. What we are doing is we are incorporating methyl group into homocysteine so that it can be converted into methionine. Methyl donor here is folic acid. Reaction is taking place in the presence of B12. So folic acid and vitamin B12 are actually interrelated in this. Deficiency of B12 will lead to folate trap. Very, very important reaction. This we are going to discuss later. But here what I am trying to tell you is because we are here dealing with the amino acids. So you can see that methionine, the most important function of this methionine is it is donating this methyl group. And this, it is going to form so many important products also. Just say for example, you can say glutathione, yeah, you can say creatine is there. You know, glutathione and creatine, these are the two very, very important tripeptides which are synthesized. Okay? So creatine, what creatine requires? It requires methionine as one of the major amino acids in it. So obviously it is donating this methyl group. So this is how it takes place. Now This homocysteine plays a very, very important role. It is nowadays considered to be a marker of CHDs also. There is an increase in the concentration of homocysteine in atherosclerosis. Now see, this is how this methionine is metabolized. I told you methionine is being converted to cysteine also. The precursor of cysteine is homocysteine here. This homocysteine is now going to combine with, very important reaction again, this homocysteine will combine with serine. There will be a combination of this condensation forming cystathionine. And the name of the enzyme is cystathionine beta synthase. Once this cystathionine is produced, this cystathionine will split up, giving out cysteine on one hand, and it is converted to form other products. So this is how this cysteine is produced. The name of the enzyme is cystathionine hydrolase, because it is splitting it. This is how cysteine is produced. Now again, once cysteine is produced, again it won't be retained. There has to be some other important products which can be synthesized from this cysteine. So what happens is, this cysteine, two molecules of this cysteine combine to form cysteine. So this cysteine can be converted into cysteine also. The name of the enzyme is cysteine reductase. On the other hand, this cysteine is also required for the synthesis of taurine. Taurine, which is one of the very, very important in bile acids. And this cysteine, once its function is over, it is converted into cysteine sulfinate, which degrades to form pyruvate plus ammonia plus hydrogen sulfide in the presence of enzyme cysteine sulfinate oxygenase. So this is how you can say, mix the metabolism of methionine and cysteine. Methionine being a essential amino acid because it is required for the synthesis of cysteine, and methionine is a methyl donor. It is giving one methyl group to form you can say form so many important compounds. Then last segment in this is, what are the inborn errors of metabolism associated with methionine. Now first inborn error of metabolism is cystinuria. There is accumulation and excretion of 4 amino acids in urine, which is also known as COLA: cysteine, ornithine, lysine and arginine. And the reason is there is defective reabsorption of cysteine. Then another is homocystinuria type 1. This homocystinuria type 1 is due to the deficiency of cystathionine beta synthase. And third is homocystinuria type 2, which is due to homocysteine methyl transferase. So these are the 3 inborn errors of metabolism associated with methionine and cysteine. And methionine and cysteine are the 2 sulfur containing amino acids. Now then we come over to another important metabolism the branched chain amino acids. Branched chain amino acids. BCAA. There are three branched chain amino acids: valine, leucine, and isoleucine. Now, if we see the metabolism of these three branched chain amino acids, three steps are common to all of them. And after that, then their fate is decided, whether it is glucogenic, whether it is ketogenic, or whether it is acting as both. Now, you can say, let's take up the similar steps. Obviously, these are the amino acids. They will undergo transamination first. So you can say first reaction would be transamination. Right. If this valine undergoes transamination, it forms alpha keto iso valerate. If this leucine undergoes, it forms alpha keto iso caproate. If this isoleucine undergoes transamination, it forms alpha keto beta methyl pronate. Transamination, we know it requires PLP as the coenzyme. Without PLP, there won't be any transamination. And this transamination, actually, see, branched chain amino acids during starvation, their degradation will increase in the muscle. But when in the well-fed state, their degradation will increase in the liver. This is, you can say, differentiation between transamination, which is taking place in the muscle as well as liver. Now, once they undergo transamination, they undergo a very important process, which is oxidative decarboxylation. I'll write it here. Oxidative decarboxylation. Now, this oxidative decarboxylation, the name of the enzyme is alpha keto acid dehydrogenase. This is just similar to PDH. It requires TPP, it requires lipoic acid, it requires NAD, it requires FAD. So this is actually an example of multi-enzyme complex also. So second step is oxidative decarboxylation. In this oxidative decarboxylation, it is going to form isobutyryl-CoA. This is going to form isovalyryl-CoA. And this is going to form alpha-methyl isovalyryl-CoA. Right? Yeah. So these are the two important reactions. Third reaction is dehydrogenation. So they will be converted back into methyl CoA. This will be converted into beta methyl crotinyl CoA, and this will be converted into tiglyl CoA. So you can say these three amino acids, branched chain, valine, leucine and isoleucine, will undergo three common steps: transamination, oxidative decarboxylation, dehydrogenation, forming the respective compounds. Now these are the common steps. Then their metabolism is accordingly decided. Now what happens is this valine will form, this valine is going to form propionyl CoA. this leucine is going to form HMG-CoA and isoleucine is going to form acetyl-CoA and propionyl-CoA. So you can say this valine is forming propionyl-CoA, this leucine is forming HMG-CoA and isoleucine is forming acetyl coenzyme A and propionyl-CoA. So if we see here, it is clear, since valine is forming a propionyl-CoA, this is strictly a glucogenic amino acid. HMG-CoA, you know HMG-CoA splits up into acetyl coenzyme A and acetoacetate. So leucine is a strictly a potent ketogenic amino acid and isoleucine is giving acetyl coenzyme A and propionyl-CoA. So this is what? This is glucogenic as well as it is ketogenic in nature. So out of these three branch chain amino acids, valine is glucogenic, leucine is ketogenic and isoleucine is glucogenic as well as ketogenic. Now this is how we study the metabolism of branch chain amino acids. Then we come over to the inborn error of metabolism. Now, inborn error of metabolism, the most important inborn error of metabolism is maple syrup urine disease. Very, very important. Urine disease. The enzyme is this alpha ketoacid dehydrogenase, or it is decarboxylase. So this is one inborn error of metabolism, and second inborn error of metabolism is isovalerial acidemia. The deficient enzyme is isovalerial dehydrogenase. So these are the two inborn errors of metabolism associated to branch chain amino acids. Now see, why this is known as maple syrup urine disease? Because the urine starts giving the smell of burnt sugar. That is why it is maple syrup. And this is how we study the metabolism of the branch chain amino acids. So by now I have finished the metabolism of phenylalanine, then tryptophan, then sulfur containing amino acids, and last but not the least, it's branch chain amino acids. Then in the next class, I am going to take up the metabolism of different other amino acids like glycine. Similarly, arginine is there because there are 20 standard amino acids. Out of these 20 amino acids, you can say 8 are essential, 2 are semi-essential and 10 are non-essential. So far we have discussed the metabolism of these. In the next class, I am going to finish up with the entire topic of metabolism of amino acids. Thank you so much. Have a good day.